So in this video, we are going to be talking about prokaryotic cells, which can be broken up into two categories, archaea and bacteria. Originally, archaea were thought to be bacteria because they possess similar traits, but they ended up getting their whole other category once they were studied more. So in other videos or classes, you might have seen the comparisons between eukaryotic and prokaryotic cells. So this video won't be focusing specifically on that, but it's important to mention that prokaryotic cells are much more simple in nature than eukaryotic cells are. For example, they have much less organelles. They're typically much smaller. They were around before eukaryotes. And another important feature to note is that unlike eukaryotic cells, prokaryotic cells do not have a nucleus. Instead, they have what's called a nucleoid, which is an area where DNA is condensed. And this does not mean that the DNA is anywhere in the cell. Like, it's still very highly organized. It's just not to the extent of the eukaryotes. So something to know about prokaryotes, all bacteria are prokaryotes, but prokaryotes can be bacteria or archaea, like we mentioned earlier. Prokaryotes can be found in all different types of environments, from extreme temperatures, extreme environments, to just somewhere that we would consider regular. Like mentioned before, typically smaller, lack a nucleus. Also, prokaryotes are usually single cellular organisms, and they can only produce via asexual reproduction. All prokaryotes have a cell wall, which has to do with the structural integrity of the cell. They all have cell membranes, which allow things to come in and out. Cytoplasms, which is the stuff that fills the cell and suspends the organelles within it. DNA, which is contained in the nucleoid. And the nucleoid of prokaryotes usually contains a single circular chromosome. So in eukaryotes, they usually have many linear ones. And some prokaryotes do have more than one chromosome, but usually it's just one circular one. And they also all have ribosomes, and ribosomes are the site of protein synthesis. They're freestanding structures, and they're distributed throughout the cytoplasm. It's also important to note that some prokaryotes will have capsules, which is like an outer protective layer, plasmids, which are small circular structures that will also contain more DNA outside of the nucleoid, flagella, which helps with locomotion, it helps them move around, and pili. Those are basically just the small hair-like projections you can see in this diagram coming off on the outside of the cell that help them attach to different things that they come across. So cells can be broken up into three main orders, as mentioned earlier: bacteria, archaea, and eukaryota. So in this video, we're talking about the prokaryota organism, or cells, which bacteria and archaea fall under. So in this video, we're talking about this circled area right here, and the blue and the red. So what do prokaryotes do? So typically when you hear the word bacteria comes to mind. You think of things that are going to make you sick, or things that make your milk go bad so fast. But honestly, bacteria, prokaryotes in general, they have a lot of good things. So stop thinking about them as being evil and start realizing they're very important in ecological cycles. They work as the primary decomposers. They're essential to many ecological cycles, including the carbon and nitrogen cycles. They give you awesome food like yogurt. They're good for you. Antibiotics can be made from bacteria to protect us from other harmful bacteria. You can also manipulate certain types of bacteria into producing things like insulin, which is awesome. The prokaryotes also will live in your intestine, which is kind of weird to think about, but they do help us break down and absorb nutrients that we need in order to survive. So those bacteria that live in our gut, we're living in a symbiotic relationship with them. They benefit from us. We're feeding them and giving them a place to live, and we're benefiting from them because they're letting us absorb nutrients we wouldn't normally be able to. Another thing prokaryotes do is, specifically bacteria, is they prevent pathogens from entering our body, um, which is awesome, and they can also be used as pesticides to kill like unwanted species of bugs on crops. So on this slide, the yellow indicates the ways that prokaryotes reproduce, and the green is indicating other ways that genetic information from prokaryotes can be transferred. So as mentioned before, prokaryotes reproduce asexually. More specifically though, they reproduce through binary fission. Binary fission just means that they replicate their DNA, and then the babies or the daughter's cells that come off of it are identical to the parent. So there's really no genetic diversity there at all. Some genetic diversity can occur through these like highlighted green terms. So transformation is basically the prokaryote intakes DNA from other prokaryotes in the environment. Transduction, which is basically when the prokaryote is infected by a virus and the virus injects different pieces of the DNA that it had picked up from other prokaryotes. The last one on the slide is conjugation, which is basically just saying that the DNA can be transferred between prokaryotes directly through their pilus, or pili, I should say. And there's some diagrams to the left on the slide explaining, so the top one's binary fission, it shows them just splitting into two identical cells, and then the lower one shows transformation, transduction, and conjugation, just to give you a visual there. So the differences between archaea and bacteria, we're going to get into this next. Here are some different structural configurations for each of them. So bacteria typically live in all types of environments. Archaea, though, are known as being extremophiles. They're living in like crazy places like deep sea trenches, they're living on tops of mountains, living in hot springs, typical areas where other organisms aren't thought to survive. Bacteria, they use glycolysis in their Krebs cycle in order for their metabolic pathways for glucose oxidation. And archaea don't use glycolysis or Krebs cycle, they use alternative pathways. Bacteria, many of them perform oxygen-generating photosynthesis. Archaea never performs the oxygen-generating photosynthesis. They can use light, but they're not producing oxygen from that process. So it's a little bit different than just like the normal photosynthesis that comes to mind when you think about that. Bacteria, typically, they're divided into categories, gram-negative versus gram-positive. Those can be categorized through staining. And then archaea are categorized through other characteristics. So those are three of them listed right there. Bacteria has thymine and tRNA, and interestingly enough, archaea does not have thymine and tRNA, which is transfer RNA. In bacteria, introns are absent, where in archaea, introns are present, and introns are just the non-coding regions in DNA and RNA. And the last important thing to note is that bacteria can be pathogenic or non-pathogenic, whereas archaea are non-pathogenic, which just means like archaea are not disease-causing, ever. The bacteria can be disease-causing or they can be not disease-causing. So in this video, we talked about the different categories of prokaryotes, and I hope that helped for clarification. Thank you.